Welcome to today's webinar on the classification of medicinal products, ATMPs, and medical devices. We will be looking at these different categories, number one, from a general perspective, and number two, mostly from the perspective of the product that we are developing here at iPSpine, which, as you know, contains a smart biomaterial and a cell component with induced pluripotent stem cells for spinal regeneration. So we will be looking at any options that we have for classification of this particular product and the questions that we have to ask in order to find the pathway that we're about to follow for the classification of this product. So mind you, I will be providing the questions, but the answers will be up to you eventually. In this talk, we have the following agenda. We will be starting with an eagle's eye view with a very high dimensions, looking at the concept of medicinal product and medical device, what these two categories are and how they're differentiated. And we will, of course, find out where to allocate ATMPs within these categories. We will then distinguish ATMPs from other product categories that are similar looking and find out what is special about them. And then we'll look at different subtypes of ATMPs. Since all of this is somewhat theoretical, we will also have some examples from real life. So luckily the CUT, which as you know is an EMA committee, has a wonderful habit of publishing their classification examples. So whenever they had a classification exercise, they publish a short summary. So we will take a look at some of these summaries of similar looking products and see how these different terms are interpreted for actual products. Once we figured out what ATMPs are and what types are available, we will find out that there is yet another option, which are the combined ATMPs. So we will talk about these next and discuss what the role of the matrix component is critical here. And then we will be zooming in on the detail level and look at the different components of our ATMP, especially at the components of the cell and the matrix part. So we will figure out what makes these components active substances, starting materials, raw materials, or excipients, which we also need to define during our classification exercise. Again, in the end, we will have some examples from real life that we get from the EMA homepage from the CUT classification exercises. And we'll finish off with a couple of take-home messages that will bring up everything in a nutshell. So starting off with the very general concepts, we will look at what medicinal products and medical devices are. I would like to call them the yin and the yang of medical science because they're these two opposing principles, mutually exclusive by definition, that together make up this great entity of means to alleviate human suffering, I believe. So by definition, a medicinal product is a product or a means to diagnose, prevent, or cure human disease by a pharmacological, metabolic, or immunological mechanism. So by the way, there is always this inclusion of a diagnostic and preventive tools. Since we're looking at the therapy today, I will probably omit this definition sometimes and just talk about therapeutic or curative approaches. Keep in the back of your mind that diagnostic and preventive tools are always included in these definitions. So we have the medicinal products that act via this pharmacological, immunological, metabolic means. On the other hand, by definition, a medical device is anything to diagnose, prevent, or cure human disease via a non-pharmacological, metabolic, or immunological mechanism. So I think it's a very neat definition because it will make sure that whatever mechanism you will ever come up with, it will always be covered somehow by these two definitions. In real life, the situation is much more simple and more straightforward. It turns out that medical devices are always or almost always acting through a physical or mechanical mechanism. So medical devices are a very diverse group, so they can be anything from wheelchairs, mirroring spoons for medicines, artificial heart valves, or bone cement. So anything where you have this physical action in the foreground is a medical device, whereas medicinal products are always something pharmacological, metabolic, immunological that enters the body and modulates the cell-to-cell signaling. But we will have some examples of both types later on. If we look at the regulatory landscape, you can see again these two separate universes, the white medicinal products on the left and the black medical devices on the right. If you're interested in further details on the regulations, you should watch the webinar of my colleague Jacqueline, which will also be online soon. I will not go into detail here. I will also not read the name of the different regulations and directives to you because it's kind of tedious. I will sometimes quote from these. When I do so, the name of the directive and the source will be on the bottom of the slide. So should you be interested in further reading, you can always look up where the quotation came from. So in this picture, I would like to draw your attention to two details, apart from the fact that medical devices and medicinal products are totally distinct universes, as I already said. Number one, we can find the ATMPs easily and very clearly within this white bubble of medicinal products, which also makes sense, I think, because as you probably know, and as we'll find out in a moment, they often have living cells and living tissues as part of their active substance, so they will always somehow signal and modulate the human body. So this is certainly nothing that is simply mechanical or physical. The second thing I would like to point out here is that well, there are other subgroups of medicinal products, and especially there is one which is the human tissues and human cells, also covered by a separate directive. These do overlap with ATMPs, but but not completely. So these are two distinct groups overlapping, but not identical. So this is what we are. The first question we're going to ask: what is the difference between an ATMP, or actually the group of cell-based ATMPs? We'll get to that in a minute. Between any other human tissues or human cells. So what is the distinctive feature? And there are two magic words here. Number one is substantial manipulation, and number two is non-homologous use. So if either or both of these criteria apply, we will have an ATMP. So what is substantial manipulation? By definition, this is a manipulation where the biological characteristics, physiological function, or structural properties that are relevant for the intended use have been altered. And we'll get to examples in just a minute. Now, altered function or non-homologous use is defined as cells or tissues are not intended for the same essential functions at the donor and the recipient site. So this means even if you do not substantially manipulate your cells to this end, if you put them to some other place where they do something totally different from before, you will also be considered to have an ATMP rather than just a cell product. So maybe one example of a non-ATMP, sometimes people experience bone loss, for example, in the jaw after some dental inflammatory condition, and very frequently a little piece of bone is taken from the thigh or some other place and inserted into the jaw. In this case, you had a bone before, it's a bone afterwards. It was not really manipulated. You just transplant this piece of bone to another site. So this is not an ATMP. An ATMP always means that you've done something substantially changing the cells to do something different. So, by the way, ATMP stands for Advanced Therapy Medicinal Product. So it is always something, you know, more fancy and more elaborate than just transplanting and transferring a tissue from one place to the other. Okay, so number one, what is substantial manipulation? It is none of the following. There are quite a few examples listed in the guideline. I won't read the whole list, but cutting, grinding, shaping, separating, freezing, sterilizing, all of this will somehow process the tissue, but it will not substantially manipulate the function or make them something different. So this is the list from the Directive on Medicinal Products. Some slightly more interesting examples are provided in the reflection paper on ATMP classification, which is specific to this classification exercise. So they also say enrichment for particular cell types is not a substantial manipulation. So even you have this wild mix normally in real life and you just take out some cells, they're not changed in their characteristics. Then interestingly, enzymatic digestion provided that you retain some functionally intact units. And the example given here is if you have pancreatic islets. If these small units are retained and this little bit of tissue is still preserved, then it's also not regarded substantial. And also if you radiolabel particular cell types in diagnostic circumstances, then this will also not be regarded as a substantial manipulation. On the contrary, there are also some positive examples of what will be regarded as substantial. I think the most interesting example here is the first, which is expansion in culture. So this is something that I personally would not have regarded as very substantial because you just augment the number of cells, I would have thought. But the agency explains this and says that, well, the induction of proliferation in cells not normally proliferating at this pace is an interference into the cell's normal behavior, as well as repeated attachment in detachment cycles for adherent cells. So keep in mind that this culture expansion is regarded as a substantial manipulation. This makes many products, ATMPs, also in the examples that we'll see later on, where you might not have been so certain whether this was substantial or not, but as soon as there is this expansion step, you will be in the ATMP class. Also, substantial is enzymatic digestion if you do not preserve any small units, because if you have the tissue architecture destroyed and the functional interactions between the cells, this is also regarded as a relevant change. More understandably, I believe, is cell activation or cell differentiation. This quite clearly is a substantial step, and also genetic modification, of course. So now, since we figured out how to distinguish ATMPs from other products, let's take a look at the 3 different types that are available. We will completely ignore the first type, which are gene therapy medicines, because this is definitely not what we have here. Even if we decide to genetically modify our stem cells, which is under discussion, this will be regarded as a substantial manipulation, yes, but not as a gene therapy, because gene therapies are means where you replace a defective gene in the patient by a suitable vector or something. And this is not what we're doing. So our problem in spinal regeneration is not that there was some genetic defect predisposing the individual and we're replacing the gene. Instead, we're replacing the tissue, as you all know. So we will leave those at the side for today and just look at the 2 other types, which are sometimes summarized as cell-based ATMPs. So these are the somatic cell therapy medicines and the tissue-engineered medicines. By the way, I have this very elaborate color code in this presentation to guide you through it. So whenever you see something blue, it will always relate to the somatic cell therapy. And when you see something red, it will relate to the tissue-engineered medicine. So let's take a look at the definitions of these 2 products. Number 1, the somatic cell therapy medicinal products. So the definition says that these products contain cells or tissues that have been subject to substantial manipulation or that are not intended to be used for the same essential functions in recipient and donor. This is no great surprise because I just told you that this is the definition for both types of cell-based ATMPs. So this is maybe not a great surprise. Part 2 of the definition says that these products are presented as having properties with a view of treating, preventing, or diagnosing a disease through the pharmacological, immunological, or metabolic action of its cells or tissues. This is nothing new either. If you remember, this was exactly the definition that we had for medicinal products in general. So this applies to both the somatic and the tissue engineered products. So I personally found this definition quite disappointing because it does not really give any clue to the distinction from the tissue engineered medicines. So let's take a look at the second group, the tissue engineered products. Part A of the definition says it contains or consists of engineered cells or tissues. So first of all, you think, wow, this time the cells are engineered. So let's take a look what engineered means. And interestingly, this means substantially manipulated and or not used for the same essential function in recipient and donor. And if you recall, this is exactly the definition we just had for both types of cell-based ATMPs. So we did not really learn anything new either up to now. But now, luckily, we also have part B of the definition, which finally gives us some clue on the differences between the two product types. So the tissue engineered product is administered with a view of regenerating, repairing, or replacing a human tissue. It is also said that it may contain additional substances, such as biomaterials, scaffolds, or matrices. And it is said that the cells or tissues may be viable or non-viable. By the way, non-viable tissues are extremely rare. They do exist. I even dug up an example once, but usually you have living cells for both products. So now let's finally try and compare these two types of cell-based ATMPs and see where the differences lie. So the first two lines of this table are the part that is equivalent. So both products contain cells subject to substantial manipulation with additionally or alternatively an essential function between recipients and donor. Now, the first point is not an actual difference totally distinguishing these two groups. It's just the wording is different. In the tissue engineered products, we have an explicit mention of additional components, such as a matrix or a scaffold, which is not explicitly described for the somatic products, although it's somehow implicitly described as well. So a somatic product contains or consists of cells. If it contains cells, it means it also contains something else, which may be a matrix. So it's not a clear difference, it's just a different sort of focus here. Then difference number two is not that great either. For tissue engineered products, we may also use non-viable cells, while the somatic products only have viable cells. But as I said, the non-viable cells are the exception here. The greatest point is the use of the product that is advertised in the directive. The tissue engineered product regenerates, repairs, or replaces a tissue, while the somatic product acts via a pharmacological, immunological, or metabolic action. So this pharmacological, immunological, metabolic action is also implicitly present for the tissue engineered product, which also falls under the category of medicinal products. Still, we have this exclusively pharmacological sort of action via some kind of biochemical pathways for the somatic product, whereas the tissue engineered product has this function of regeneration and replacement of defective tissues. So if we try to put this into some kind of picture to make it maybe easier to understand and to keep in mind, you may think that the somatic product is something like an external steering committee, which is implanted into the body to boss everybody around. So this is the sort of product that will instruct the body to do as it should in order to heal. So this will be products which secrete factors to induce migration or differentiation of the patient's own tissue, which might reduce inflammatory conditions just by providing the correct cues to the patient. So this is really this instructive sort of role. The tissue engineered product, on the contrary, is the cure itself. It will not provide the cues for the body to cure itself, but it will represent the novel tissue to repair the damage. So you will insert some new tissue, and this tissue will have all the properties required for the patient to get better. So you will have this filling of the gap of the defect by this tissue, and this will be the cure rather than just this modulatory action that we had for the somatic product. In this context, I think two of the above differences also make more sense. Number one, the presence or the potential presence of non-viable cells in the tissue engineered product. You can imagine that you can still stuff a hole or heal a defect by stuffing some dead cells in there. You would never be able to modulate the body's environment with a dead cell, on the contrary. And the even greater difference, the greater point here, I would say, is this explicit mention of the matrix. So if you have a product that is meant to replace a tissue, and where you think that, you know, the cells as the distinctive component making your ATMP and ATMP, if they don't do this alone, you may wonder whether it's allowed to use a matrix, which it is. But you can imagine that the guidelines sees a need for justification here to explain why it is okay to have a matrix to do part of this job of replacing this tissue and of filling this damaged structure. For the somatic products, it is also allowed to have a matrix, but in this case, it would not be so important. So you don't necessarily see a need to talk about this. So if you have these cells secreting beneficial factors, they may sit on a matrix, why not? So sure, if this helps them in being inserted in the body and remaining where they are, but you wouldn't discuss it extensively. So when you have a product where the matrix has a very pronounced and important role, you typically end up with a tissue engineered product, although it's not forbidden for the somatic ones. So I hope this became clear now. One word of caution, nevertheless, in the end, the world is typically not black and white, and it is not blue and red either. So it's very difficult very often to draw a clear line between these two product types. And many, and I believe also our product, have some features of both products. So most tissue engineered products will also have some pharmacological or immunological action, talking or cross-talking to the patient's own tissues, whereas the somatic products often also will, to some extent, maybe cover a tissue defect. The question is also, is always where to place the focus, and if you try to explain the mode of action of your product to somebody, where would you see the major role? Is it more this repairing, replacing function, or is it more of the modulating function? But very often both features apply somehow, and it also depends on how you present your product, which tests you do, and which functions you want to demonstrate to see that it's efficacious. So keep in mind, it's not always clear to draw this very clear line. Also, sometimes the cuts decisions are a bit surprising, because there's always a little bit of both. But try to figure out what you think the main activity is and what the main characteristics of your product are. And now let's walk on to some actual examples from the EMA homepage. So you can find the link on the bottom of the slide where to find all of those examples. So here we see some features of the product which was presented in the classification meeting. The active ingredient are engineered matrix proteins produced by human fibroblasts. which were somehow cultivated on a polymer scaffold in vitro. The finished product is composed of this complex mixture of extracellular matrix proteins with a little bit of residual polymer, which is somehow supplied to the patient. And the proposed indication is treatment of congenital heart defects. Now try to think briefly before I show you the solution, what it is. Is this an ATMP or not? And as you probably correctly figured out, this product does not contain any cells or tissues. The cells were just used for the production of this matrix. And therefore, it was decided this is not an ATMP. Yes, and so we are already done with the classification here. Now, example 2, I will give it away already. We do have an ATMP, but of course we want to find out what type it is. So in this case, we've got autologous cells from adipose tissue, which will be supplied as some kind of suspension, and they're used for the treatment of pain associated with joint osteoarthritis. So in their justification for that decision, the agency said the product is composed of manipulated cells from stromal vascular fraction, which means, of course, that we have an ATMP here. We do not know how the cells were manipulated. They were probably expanded in culture. And likely they are also put to non-homologous use. This is not stated by the agency, but I would believe so, because adipose tissue is normally not involved with pain or inflammation. So this is likely also some kind of non-homologous use. Now, importantly, the agency said that the treatment of pain with this product is associated with a immunological action. And hopefully the word immunological rings a bell now for you, as well as the blue color, because as you remember, any product acting through an immunological mode of action is a somatic cell therapy. So this product was classified as a somatic cell therapy product. Now the next example is again an ATMP. In this case, the active ingredient is twofold. It consists of autologous mesenchymal stem cells, culture expanded. So hopefully again this will ring a bell for you. As soon as they are culture expanded, they are substantially manipulated. So we do have an ATMP. And they also have osteogenic differentiation induced in culture, which is the second good reason to talk about an ATMP. And these are embedded in a three-dimensional structure of a demineralized bone matrix. And in the finished product, we have this wonderful, and I think it really sounds very great and sophisticated. We've got this artificial tissue-like combination of these osteogenic cells that have been differentiated in vitro in this demineralized bone matrix. So a very beautiful product, which will be used for the treatment of bone defects. So let's see what the agency thinks about this product, or maybe you think first what you would like to classify it as. The agency said, well, the cells are viable and have been subjected to various treatments, including cell culture. So they are certainly substantially manipulated. We have an ATMP. And regarding the mode of action, it is stated that the treatment aims at regenerating, repairing, or replacing a tissue. So number one, these words, number two, this color very clearly indicates, hopefully, that we have a tissue engineered product here. Yeah, this is the second type of ATMP. And now we already come to the first set of take-home messages. So what we learned in this first part of the webinar was that we have medicinal products which are distinguishable from medical devices by their action. Medicinal products act via a pharmacological, immunological, or metabolic action, while medical devices typically have a physical or mechanical mode of action. We found out that ATMPs are medicinal products, and that there are two types of cell-based ATMPs, both containing substantially manipulated and/or non-homologously used cells. The distinction is that the somatic cell therapy products act via this pharmacological, immunological, metabolic action, which is characteristic for all medicinal products. So they do instruct other cells, they instruct the body to heal itself in a way by providing the correct cues. Whereas the tissue engineered product, while it also falls under the medicinal product definition, focuses more in terms of its mode of action on the regeneration, repairing, or replacing of a tissue. So these are more products that fill the gap and that represent the health tissue themselves rather than just instructing the patient's own tissues. Okay, now since we finally figured out what types of ATMPs are available, I already warned you we will find out that there is yet another possibility, which is the group of combined ATMPs. So as you can see here, combined ATMPs occur in all three classes. We've got combined versions of the here ignored gene therapies. We've got combined somatic cell therapies, and we've got combined tissue engineered medicines. The combination process as such is the same for all three groups, so I will not distinguish between these three types now, but just talk about the combination process for all three of them. So if you remember this slide from the very beginning, I said we have two different universes for medicinal products and medical devices. They are non-overlapping by definition because they've got these exclusive definitions, mutually exclusive definitions, but you can link them together and you can use both types in one product, in which case you will end up with a combined ATMP. By definition, a combined ATMP must incorporate as an integral part of the product one or more medical devices. And this is important, the device should be used in the combination in the same way as its intended use without additional components. So typically you certify the medical device separately first and then incorporate it into the combined ATMP. So if you have a medical device that is certified for vascular injury, and then you apply it in a combined ATMP to support bone growth maybe, then you can't just call this a combined ATMP because vascular injury and bone growth are just two distinct uses. So in this case, you cannot just rely on your prior certification as a medical device. So let's take a look first of all on the definition of medical devices before we take a look on whether this might apply to our product. You already had the short form at the very beginning. Now the long version says a medical device is an instrument, material, or other article intended to be used for human beings for the purpose of replacement or modification of the anatomy, physiological process, and which does not achieve its principal intended action by pharmacological, immunological, metabolic means. So if you remember, that was just the precise contrast or contrary of the medicinal product definition. So if we are thinking about potentially having a combined ATMP, we will need some kind of medical device component within this product. And we actually do only have one candidate for this job, which is the matrix component, because the cells naturally will not serve as a medical device. So if we consider that the matrix might be a medical device such that we might have a combined ATMP, we would need to demonstrate that the matrix has a primarily physical mechanical mode of action. in which case you might fall into this class. So let's take a look what a primarily physical mode of action might be. It could be a filler of tissue defects. So if you say we mostly use this matrix in order to fill the space left by the degenerate intervertebral disc to stop the vertebral segments from touching each other and hurting each other or damaging each other, this would certainly be a very mechanical effect. You just place something in there in between as a buffer. It might also be used as a vehicle for injection of cells. If we say we need a certain volume, for example, to inject it properly, or to make the whole thing fluid enough. We would also have a physical mode of action. Also, if we said we need some kind of scaffolds to protect the cells from shear forces, either during injection or maybe later on when the patient moves, or to keep the cells in place. This is also physical, of course. There are also more subtle and more complex functions which might be related to physical mode of action. For example, if you induce cell migration, maybe via local pressure, or also if you induce cell differentiation, maybe through the stiffness of the membrane you used. So there are quite a few rather complex functions that you could have via this primarily physical mode of action. This is certainly not a comprehensive list. It's just a couple of examples. But should we decide that our matrix really acts via this physical rather than metabolic and physiological mode of action, we have the option to certify the matrix as a medical device and then provide a combined ATMP later on with this matrix as a medical device inserted. So in this case, we would certify the matrix as a medical device, and this is not done in email, but it's done via a notified body. We would then apply for marketing authorization of the cells plus matrix as a combined ATMP, and this will then be looked at by the EMA, or actually by the CUT at EMA. And then in the assessment of the whole product, we would have an assessment by the notified body of the medical device part, and an assessment of the whole product by EMA and CUT. And as I already said, in the combined product, the medical device must be put to the same intended use as the medical device alone. So let's take a look at examples, again, kindly provided by the CUT on their homepage. Do we have a combined ATMP or not in this example? We have an active ingredient containing adipose-derived mesenchymal stem cells combined with beta-tricalcium phosphate. So the finished product will contain this mixture of cells and this biomaterial, and it will be used for the treatment of bone defects. Now, the agency justified their decision as follows. The product contains substantially manipulated cells, likely, not likely, clearly expanded ex vivo. So we have an ATMP. It has been presented as having properties for regeneration and repair of the human tissue. So hopefully you see in front of your inner eye now the red color for regeneration and repair. So we certainly go into the direction of a tissue-engineered medicine, as you can also see by this pronounced role of the matrix. And interestingly, this beta-tricalcium phosphate is also approved for clinical use as a medical device already. So the agency concluded that we have a combined tissue-engineered product because the biomaterial is used in the same way as its intended use for a medical device. We don't know what is filed for the medical device, but it likely is also some kind of treatment of bone defects of this wonderful hard material. So if you populate this material with cells in addition, as an additional function, you still have this same primary mode of action for the medical device as you use it alone. Now, example number 2 or example number 5 here, we've got a different product containing endothelial cells in a porcine gelatin matrix, and the proposed indication is vascular injury. Now let's see what additional information is provided by the agency. I mean, the first point is, as always, we've got substantially manipulated cells. They've been co-cultured with the gel matrix. So we have an ATMP. And interestingly, the mechanism of action is based on the release of biological factors to repair vascular injury. So even though we do not have the keywords pharmacological, metabolic, or immunological action, I think it is quite clear that we are heading into the somatic direction here, because the release of biological factors means that we're instructing the patient's damaged tissue to heal. Now, interestingly, the gel matrix is a medical device which is certified for surgical procedure as an adjunct to hemostasis. So at first glance, I would say this looks very much like a combined product, because you have this separately certified medical device. And hemostasis, I would say, is also a very similar function as treatment of vascular injury. So at first glance, I think it looks very much like a product where the medical device part is put to the same mechanical, physical function of covering the vascular injury and somehow gluing this defect as in the medical device by itself. Now, interestingly, the agency goes on saying that the gel matrix not only keeps the cells in place, which would also be a medical device sort of action, but also contributes in some way, and we're not giving any details, to provide the correct signals to the cells. So it is stated that the matrix somehow talks to the cells and makes the cells within this product do what they should, probably via some kind of cell receptor interaction. It was also considered that the matrix is remodeled by the cells, contributing to efficacy. So cells and matrix talk to each other apparently in a very elaborate way, probably lots of cell matrix signaling and cell receptor interaction. So this is regarded as a non-mechanical behavior. So the medical device here is apparently just not a piece of furniture just sitting there and providing the scaffold, but it's something alive, talking, speaking, modulating. And for this reason, the agency considered that the product is used in a different way from the intended use as a medical device, where apparently the matrix bit just is put onto the injury to keep everything, well, to hold everything together. But in this case, together with these endothelial cells, it is something living and something active. So it's not a medical device anymore. And the agency concluded it's a non-combined somatic cell therapy. Although you will probably agree that we also have a certain amount of tissue replacement and tissue repairing function with repairing of a vascular injury. But apparently it is seen that the major action is the secretion of factors for the surrounding tissue, and the matrix is not just a physical thing here, but the matrix is something that also contributes to this crosstalk and signaling. So now finally, we have made our decision. Once we have answered all these questions sometime, we have this distinction between somatic and tissue engineered medicine, which you have to figure out, which will probably be a decision mostly made by the cell people, who will take a look at their primary function of the product, whether they will be more instructive or be predominantly in the repairing tissue forming area. And we'll have to make the decision whether it's a combined product or not, which is a decision mostly made by the matrix people, who will have to define whether the matrix is acting through a physical mechanism or whether it's more something in the pharmacological metabolic area where crosstalk and cell receptor ligand interaction plays a role. So hopefully we will eventually have made this decision. And once we have done so, we can look at how to classify the individual components within this product, which are two. In our case, it's the matrix and the cells. and the main questions are to be asked for the matrix component. So how could we classify the matrix? We have two groups of options. Number one, as just described, you could say the matrix is a medical device if it has a primarily physical mode of action, in which case we could separately certify it as a medical device. If we decide that we do not regard it as a medical device, but part of a medicinal product, there are four basic options. It could be an active substance, a starting material, a raw material, or an excipient. And now we will walk through the definitions of all of these terms. Number one, active substance. I do not think I need to explain that in depth. It's quite clear, and I think the most concise definition was provided on the email homepage. It is a substance responsible for the activity of a medicine. So nothing to add here, even if you can talk about this longer. What is a starting material? So a starting material, by definition, is material from which the active substance is manufactured or extracted. And for biological medicinal products, this means a substance of biological origin, so the origin has been an organ or tissue, a cell or a fluid. Now, in the GMP guide for APIs, there is some more information. It says that an API starting material is something used in the production of an API that is incorporated as a significant structural fragment in the later API. This is important, keep that in mind. Should have defined properties and so on and so forth. But a starting material is something that will later on be provided as part of the API. This is in contrast to the raw material, which is used in manufacturing or extracting of the active substance, but from which the active substance is not directly derived, such as media, cell culture, reagents, and so on. They've been in contact with the active substance. They may also be present in traces in the end, but they are not part of the active substance later on. So if we want to differentiate these two terms against each other, as already said, the starting material is present in the final product, while the raw material is not. For our product, this means if the matrix was used for the ex vivo differentiation, and later on the cells are flushed out of this matrix and inserted all by themselves into the patient, then the matrix would be a raw material because it's used for production, but it's not present afterwards. If, however, and I think this is what we are aiming at, the matrix is used together with the cells and will also be implanted along with the cells into the body, it is a starting material because it is present in the final product. Now the last option that we have is that it could be an excipient. An excipient is anything but the active substance and the packaging material of medicinal product. EMA homepage says it is added to the medicine for a specific purpose. It's always added for a specific purpose. It's not just an impurity that arises without intention. It can be of human or animal origin. So if you want to define what our components really are, I think it's obvious that the basic question we need to come down to is what is our API? As soon as we know what the active ingredient is of the whole product, we can find out what the different components are. So let's take a look at the different possibilities. Again, we've got a very elaborate color code, by the way. The cells are yellow, the matrix is green, and the API is pink and is denoted by this pink circle on the left. So if we decide that the API responsible for the therapeutic activity is the cells, then very clearly the matrix is merely supportive, in which case it would be an excipient. In our case, it's a novel excipient because we're just developing it, so it has not been used for this end before. So we very clearly have cells all alone as active substance, and the matrix is somehow in the background just supporting it. Alternatively, we could say, well, our API is twofold. It consists of two components. Component 1, cells, component 2, matrix. In this case, both cells and matrix are required for the therapeutic activity, and they would both be starter materials. It might be called intermediate material for the cells, but that shouldn't worry us now. But we would have a starter material for the matrix because it's part of the API, and the API is made up of it, but it's not the API alone. A third option is more of a theoretical possibility. We could say that both the cells and the matrix are an active substance in their own right. And we combine them because there is some synergistic activity that further enhances therapeutic activity. We will not do that because it's very complicated. It will result in a lot of additional testing that we do not feel like doing. But theoretically, this is also possible, having two APIs. Now, once more, and for the last time, let's look at some examples from the email homepage. So example number one, we've got the matrix as excipient in this example, which is also very easy to find on the definitions. The active ingredient is cultured autologous chondrocytes. So very clearly, it's just cells that make up the active ingredient. The finished product has this active ingredient in a fibrin-based excipient of human origin. So it's already given away here. The fibrin part is an excipient. And the indication is treatment of some kind of cartilage defects. So what did the agency write? They wrote the cells are engineered and substantially manipulated. They have been cultured somehow. And they're administered with a view to regenerating, repairing, and replacing a human tissue, this cartilage defect. And as you all guess already, and you see by the red color and by these keywords, regenerating, repairing, and replacing. We have a tissue engineered product here with an excipient, which is the matrix component here. Now we get to my favorite example. This is again example number 3, which we looked at previously. You remember we had these autologous mesenchymal stem cells, culture expanded and differentiated to an osteogenic phenotype in a demineralized bone matrix. So we've got this wonderful artificial tissue consisting of cells in this bone matrix structure for the treatment of bone defects. I won't repeat what we just heard. We've got a treatment regenerating and repairing a tissue, repairing this bone by this wonderful demineralized matrix with the cells. So we've got a tissue engineered product here. Now, interestingly, this is not combined. You remember we previously had an example with tricalcium phosphate mending the bone structure just like it does without the cells. And there it was considered that, well, never mind what this one was. But in this very case, anyway, we have a situation where the demineralized bone matrix is used as part of the active substance and not just as something acting like a medicinal product and providing the physical structure. This was justified very much in detail by the agency, so I can really recommend reading this example. It's very interesting on the way they think. Part 1 is just background that we know from the beginning of this talk. The cells contain viable cells. They've been substantially manipulated. They regenerate or replace or repair a tissue. So we very clearly have an ATMP, and we very clearly have a tissue engineered product. Although it is also stated that this combination promotes angiogenesis and osteogenesis. So if you remember, hopefully from the beginning of this talk, it's sometimes not so easy to draw a clear line between the somatic and the tissue engineered product. And this is a case where we say, okay, the primary action is regeneration and replacement of this tissue. But in addition, we also have features of signaling to the surrounding tissue. We've got angiogenesis and we've got osteogenesis. So we do also modulate the surrounding tissue somehow. But it was considered that the primary activity of this product is this replacement of a tissue. So it is called a tissue engineered product, although it has some features of both products. And now the interesting bit is how does the agency argue that we do not have a combined ATMP? So this is because, as you see in the second paragraph, the demineralized bone matrix is biologically active. It potentiates the osteogenic phenotype, so it does talk to the cells, make them do as they should. It induces collagen synthesis. It's not a medical device. It talks to the cells. It is something active. It is something living. It's not just some physical support thing. So this is why the agency said here, this is not a combined ATMP. This is a tissue engineered product where the matrix is part of the active substance, and therefore it will be considered a starting material. So we come to the take-home messages number 2. We found out that combined ATMPs contain a separately certified medical device. If we were to classify as a combined ATMP, we would need to demonstrate that the matrix is a medical device or can be classified as such because it acts via a primarily physical mode of action. If we decide that we have a combined ATMP, the matrix will be a medical device, the API being the cells. If we decide that we do not go into this direction of the non-combined ATMPs, the matrix could be an excipient, in which case the API would exclusively be defined by the cells, or the cell could, the matrix could be a starting material, in which case the API would be made up of cells and matrix. So a wrap up of this whole talk, we started off with this eagle's eye view of product classification. We figured out how to tell a cell-based ATMP from something else. And hopefully somewhere in the back of your mind, you will hear the magic words, substantial manipulation, non-homologous use. We figured out what differentiates a somatic cell therapy product from a tissue engineered product. And hopefully in the back of your mind, you will see the blue somatic products instructing others, modulating the patient's own tissues by secreting beneficial factors, in contrast to the red tissue engineered product, which fills the tissue defect itself and will provide new healthy tissue. And we figured out why combined ATMPs require a separate certification. This is because a combined ATMP contains a medical device inserted as a substantial part of the product, and this medical device will be separately certified by a notified body. Then when we zoomed in a little bit on the component classification, we found that it's very important to figure out what the mode of action of a matrix is. If it is mainly physical, we may opt for the combined ATMP option where the matrix is a medical device. Alternatively, it could still be regarded as an excipient. If you find that the matrix does something pharmacological, immunological, metabolic, if it talks to the cells and mediates some differentiating properties through some receptor-ligand interaction, it would likely be regarded as a starting material if it is later on part of the active substance, or it might be an excipient if we say that this activity is not so pronounced and not so essential for the therapeutic function. We also found out why it is important to define at a relatively early stage what the active substance will be, because it will have an impact on what the individual components are and how they need to be tested. So if we say that the active substance is the cells only, then the matrix must be an excipient or a medical device. Whereas if we say that our active substance is composed of cells plus matrix, the matrix will be a starting material. So we've come to the end of this webinar. I hope you enjoyed it. Thanks a lot for listening. Should you have questions, comments, feedback, or wish to talk to me for any other reason, you have my contact details on the slide. So thank you very much and goodbye.